Good morning. It's 9 o'clock here in San Diego, so we will start the webinar. Thank you very much for joining us. Today's webinar is on linking protein structure to sequence and alignments. My name is Andrew Uri, and if you have any questions after the webinar, my email address is here and my phone number is here. Please call. But during the webinar, if you have any questions, I can try and answer them during the webinar or at the end. And the best way to do that is via the questions panel in your GoToMeeting dialog window on your computer. You can also follow MolSoft on Facebook and LinkedIn and Twitter, and you can watch the recording of the video on YouTube. A link will be sent out also after the webinar with the link to the recording. And also, if you want a trial version or evaluation version of the software that we're showing, please contact me by email and I can send you that. We can offer a month's license. So the key topics of today's webinar is how to read in sequence data into ICM and also extract sequences from the protein data bank from a 3D structure. Then how to make that an alignment, and then how to edit the alignment manually, tweaking the alignment, and then also coloring, annotation, making images of an alignment for publications, and then how to link an alignment to protein 3D structure, and then how to display sequence conservation in the ligand binding pocket, linking the protein structure to 3D. So ICM has a direct link to three key sites. The first is Uniprot, which is a general repository of protein sequence data and function data. To ProSite, which contains annotated protein domains, families and functional sites, and patterns and profiles. And also to the PFAM database, which is a curated database of protein families. So you can read in a sequence alignments of protein families directly from PFAM into ICM. So just a quick overview of the methods. In ICM there are a variety of alignment methods that are incorporated into ICM, so you can choose which one you want to use. The famous Needleman-Wunsch alignment method, and Smith-Waterman, Wilbur-Lippmann, and a Zager method developed by Ruben Abagayan, the founder of MolSoft and currently at UCSD. And built in ICM there are a variety of comparison matrices which you can select, Blossom, Gonnet, and different varieties of Blossom that you can choose. There's also a secondary structure prediction method, which is basically the DSSP method, a slight modification of that Kavshin-Sander DSSP secondary structure prediction method, which is based on the observed pattern of hydrogen bonds in a three-dimensional structure. So we have two main examples. The first is a kinase example where we're going to read in 3D structure, sequences, build an alignment, and then map the conservation onto the 3D structure. So it's a useful way of spotting maybe regions of the protein where you've got maybe unconserved regions where maybe you could design molecules towards that region. If you have a pocket, if you run ICM Pocket Finder and you have a selection of allosteric pockets, you may want to try and find one that's maybe doesn't have conserved regions where you can maybe develop a specific compound, so you can map this data directly onto a 3D structure. And this is completely linked, so if you make a selection here, that selection is propagated into the alignment method. The second example is setting us up for next week. 2 weeks from now, the webinar is on modeling, and a challenging set of structures for modeling are the G protein coupled receptors, and obviously they're major drug targets, so it's a useful example. They all share a common 7 transmembrane helical region, but they lack significant sequence homology between the subfamilies. And so to model these, you need to rely on key key motifs in each helix which are conserved. So for example, there's a DRRY motif which is conserved in helix 3. So we'll be annotating our alignment based on these key motifs for the homology modeling example in 2 weeks' time. You can also read from ProCite the Class A GPCR alignments directly into ICM, which helps you guide your alignment of your model and template structure. Okay, so we'll go to the example. So when you open ICM, so all these alignments are in a product called ICM-Bio and also in ICM Pro. ICM Pro is our main desktop modeling software. So we have a search tab and we have a direct link to UniProt, so we can enter a UniProt code for a structure. I'm going to enter pim1_human. This reads in the structure from the sequence from pim1 from UniProt. Okay, so we can see the sequence in the ICM workspace here. We notice that it's also read in a number of sites directly from UniProt. So it's telling us different regions of the ligand of the sequence. So it has variants mapped directly from PubMed onto this structure. So it'd be nice to map these sites directly onto the sequence. So you can see that by if you right-click on the ligand sorry, I need to move into sequence and then choose the view in alignment panel. We can see that these annotations are mapped directly onto the sequence, so we can select this is a particular the protein kinase domain. There is this one is a mutation linked here, and you can toggle these on and off depending on what level of detail you want, shown here. So these are phosphorylated residues. So you can select them. Okay, so that's one way of reading in sequence into ICM. Another way would be to you can copy and paste sequence. So you go copy a sequence from text or from a different, or from UniProt directly, or from FASTA file, and then you go File, New, and there's a Sequence tab, and you can just paste your sequence, give it a sequence name. It's just the N-terminal, and go OK. So we can now see that we have 2 sequences here. I'm just going to delete this line. So you have 2 sequences. Another way to read in sequence information would be directly from a saved FASTA format file, and I have that here. So the file is just here, so it reads in a similar way. So now we have 3 sequences. This one, you notice that this sequence we read from UniProt actually has some secondary structure annotated. We can see that underlined is the green, which means beta sheet. Red is alpha helical, and magenta and blue is sort of non-canonical helix. So magenta is a 310 helix, and if you had one underlined in blue, that would be a pi helix. But because we read this raw sequence in, it doesn't have any secondary structure associated with it, but we can use the inbuilt DSSP secondary structure method, which has been modified by ICM a little bit, to predict secondary structure for this sequence. So you right click on here and choose predict secondary structure. If you had secondary structure predicted already, you could ignore it, but we don't have it in that case. So now you can see the secondary structure of this sequence predicted here. Okay, so we can also extract sequence directly from a PDB file. So we can go to our PDB search in the search tab, the PDB search here. We can enter PDB code, PHK. That reads in this kinase structure. So far we've got 3 kinase sequences. And we can extract the sequence from this A chain here to make an alignment. So you can see that the A chain has a sequence. We can extract that by right-clicking and choosing Extract Sequence. Go OK. So now we have 4 sequences here. Just as an example, I'm going to read in another one, 1QL6. And we extract the sequence the same way, right-click, Extract Sequence, and go OK. So this little symbol here and here indicates that the sequence 2PHK_A, which is here, is directly linked to this structure. These 2 structures, the structure and the sequence, are linked, which will become in handy in a minute when we start manipulating. I'm just going to color by object just for clarity. See objects. Okay, and to complete the example, I'm just going to read in a couple more sequences from PDB, from the UniProt. CHK2, kinase, and MK14. Okay, this end terminal is not very useful. I'm just going to delete that one. So we have 6 sequences here. Okay, so to make an alignment, we can select, double click here to select them, or you can use Control, hold Control and click if you want to select a non-contiguous range of sequences, or you can choose a full range using Tab, or double click here and that'll select them. So they need to be selected. So blue means it's selected. And then we right click on the selection and choose Align Sequences. And this brings up the alignment panel here. And we also note that we now have in the structure we now have another symbol here, which means that this structure is linked to this alignment called ALN. So that means that if I make a selection in the alignment here, for example, the sequence, the selection is propagated to this structure. You see that the green crosses here for both structures, and also here. So you can see the selection here, here, and here. So anything I do in the display will be propagated to the selection. Okay. So the aim of this first example is to try and observe the sequence identity in the ATP binding pocket between these 2 kinases and see the identity in sequence as well. So it might be nice to superimpose these 2 structures. We showed that last week. You can use a rectangular selection or use your right-click button and drag over those 2 structures. Go to Display tab and superimpose. And those two kinase structures are superimposed. Like that. As I mentioned last week, they're fully dynamic, so you can superimpose anything you select, basically. So the N terminal or the pocket region, like that. It's fully dynamic. Okay, so now these 2 structures are superimposed. So to define the pockets, we have the ATP molecule here in the pocket, and we can select the residues surrounding the ATP and try and see the conservation, sequence conservation. So if we right click on the ATP and choose, you can choose neighbors and choose 5 angstrom radius, and the type of selection is we want to select on all objects that we have. So we only have 2, but we'll choose all objects. We'll exclude the source. We won't select any waters and go OK. You can see that the selection has been made here, and some of this selection has been propagated to the alignment, to the 1QL6, which is displayed, and the 2PHK sequence. So we're interested in how well conserved that pocket is compared to the other kinases that we've read into our alignment. So we have a selection here, and we can propagate this selection to all sequences. You see that we click on this, so this panel here, this button here, opens up a new panel on the right hand side, and there's an option called propagate to all sequences. So we can see once again that this is linked, and so it'd be nice to just display the residues in the binding pocket in the alignment, and so then you can directly see the conservation. So we want to hide everything else outside of the pocket, so we just have the residues here. So to do that, we have some selection tools, and you can invert the selection. So now everything outside of the pocket has been selected, and then we'll hide. Click hide. And you see now that we're left with just the residues in the pocket. So we can see that there's a sort of slightly variable region here at the back of the pocket, where it's semi-conserved isoleucine valine. We actually put a more diverse region here. It's sort of coming out towards the DFG region, which might be a good indication that you want to build if you're designing a ligand, you'd want to probably come out this region here. You can see that obviously the glycines in the glycine-rich loop on above are fully conserved. Okay, so, and you could write out this image, save to clipboard, or save as PNG image. So we can also color the structure by conservation. So this coloring is a default inbuilt in ICM, but you can change it to any coloring you want. You see above here is consensus strength. So we can change it directly to conservation. This gives, I was going to show the whole sequence. So if you change it to conservation, it's going to undisplay these slides. So 100% conserved are in dark green, semi-conserved are sort of yellow, greenish. The coloring scheme is up to you. So then if we want to propagate this coloring to the alignment, to the 3D structure, that gives us a more visual way of identifying where there's diversity in the pocket, where there's conservation. So we can, this is displayed in ribbon. So if you click and hold on the ribbon button and choose the option color by alignment, this shows the coloring. So you can see these 2 glycines conserved here, glycine in the glycine-rich loop, and this is probably the DFG region here. It's fully conserved, obviously, in the kinase, and the yellow coloring is semi-conserved regions as well. So you can change the coloring here to no color if you wanted to. There are by residue type. ICM-Bio was we developed for somebody who had this scheme in mind. So charged are blue, positively charged are blue, negatively charged red. There's other things. You can see the coloring, what defines each color. If you click on the edit button here, and you can change that yourself in this table here. So I'm going to delete this table. Okay. So I'm going to go back to the consensus strength. So you can change the strength of the color of the consensus by dragging on this bar here. So if I say 100%, that means only the residues that are 100% conserved throughout your alignment will be colored green, for example. Or you can shift this to 50%, so this gives this flags, or 0, then everything is colored. Yeah. So other tools in the alignment window are the ruler. You can show the ruler with the numbering here. You can add the tree. The tree is here. You can drag the distance to rearrange the order of the alignment. So you can manually move a sequence around the alignment by dragging it. I think you should be able to. Yeah, so when you haven't got tree displayed, you can drag so maybe for a publication quality image, when you built a model based on a template structure, you may want to drag your template structure to the bottom, for example, or maybe to the top, so you can compare with the template with your other sequences. Okay. So the other tools are, you can show just the tree only here. You can flag unaligned regions, or it's just unaligned completely. View the differences. It's just a different way of. So here we have the leucine, alanine. The alanine is the only difference. Just makes it clear. You have horizontal scroll, so everything is in horizontal rather than there's no dragging down. Okay. So this next panel is on selections. So we sort of covered some of those key things using the select, invert, and hide. Sometimes when you maybe selected one residue, it's sometimes hard to find in the big alignment, so you can highlight those selections and it start flashing, and you can find them easily in a large alignment. There are also some preferences to hide gaps. So if we want to hide this region, you can change the auto hide gaps limit. So it automatically hide gaps of 5 residues or less. And the hidden block region, you can have a length, and it will show you the length of that region that's hidden, and I'll show you here. You can also display the secondary structure. So as I mentioned before, so this is displaying all the secondary structure. You just show it for one of them. So alpha helical region, so in red, beta sheet here, and magenta is 310 helix, and blue is pi helix. Okay. So we can also propagate this coloring to, as I showed, to the ribbon, but you can also propagate it to surfaces as well, which is sometimes helpful. So we can build a surface of the A subunit of 2 PHK, go to meshes, and then choose plain solid. Okay, this builds a plain solid. And then maybe we want to select all the fully conserved residues in, so X is basically on the consensus line, so where there's fully conserved, X means 100% conserved. And so we can color the conserved residues on the surface. The surface is here. It's called G_skin 28 PHK. So we right click on the surface and choose color by atom selection, and we can choose green for fully conserved. We may also want to select just the semi-conserved one, so we can add the consensus here. Semi-conserved is a hash. So if you add a hash here, and same way choose select, so we have all the semi-conserved, and then we can color semi-conserved yellow. So you can see, we can see quite clearly that the ATP pocket is very conserved in this alignment. Okay. So the next example is using GPCRs. So this GPCR alignment is an adapted alignment from ProSite. So to make this alignment, what I did was go to File, Load, and then you can load a PFAM alignment or a ProSite alignment. And I read in the ProSite alignment for GPCR Class A proteins. So you can see that there is a sorry, trying to clean it up a little. So we can see that there's a disulfide here, and we can jump to that region. So we have disulfide between intercellular loop 2 and one of the helices. We can also read in other annotation from Uniprot. So from the ICM workspace panel. So we have a number of different sites here from the PDB that are directed here, and there's also sites that are read in directly from Uniprot. So we can also view the 7 transmembrane regions by showing the secondary structure, for example. So we can see that the first helix is here. You can select that region. And that's once again propagated to the 3D, so you can color that helix. So there's no need to go to UniProt and look for numbering or anything, because that information is directly transferred in your sequence, so you can easily select regions here. So you might want to box this region, so flag it. So it's a TM region, so we can right click and choose draw box. This builds a box around our selection. You can also annotate that region with text. So right click and choose annotate region and give it a name. So give it a particular type. You can change the coloring, the text. Okay, so TM1 is here. So if we just single click on the annotation, that selects that region, TM region. You can also show what is called the profile. This gives you an extra, this is basically, so this D is a large D because that aspartic acid is conserved throughout that alignment. It gives you a visual way of defining certain parts of the alignment. So we know that in the third TM region there's a dry motif, and that's here. You can see DRY flags it very nicely for us. So we can select the DRY and we can manually we can maybe change the coloring of that. We can change the shading, for example, here. Or we can change the font. You see it's in this region of the kinase here. Let's change the font to white. And bold as well. If you don't like what you've done, you can select that region and right click and choose clear. That'll go back to the original way of annotation. So you have to select what you want to do, and then you can shade, set font color, draw box, and annotate here. Okay. So you may want to do some other manipulate this alignment for maybe to build a model, for example. So we have a template structure, so you can shift an alignment using the cursor keys, and so you can edit it yourself manually using cursor keys. So you need to use left and right to drag the sequence along in the alignment. So the only way that the sequence can move is if you have a gap to move into. So move it along. You can insert a gap by selecting a region where you want to insert a gap by clicking and dragging down the alignment, and you should be able you can use the use this spacebar to make a gap. The spacebar will make a gap. So you can edit it however you want. You see that the consensus moves with it here. Okay. So you can also add a title to the alignment. here. So as we know that GPCRs have the same structural features, but sequence-wise they are quite diverse, and so we can see the diversity in a table. So we select this TM1, we can right-click and choose. We go to right-click on the name, and we can calculate the sequence similarity in TM1 from this alignment. So we single-click. It's asking us for the reference sequence, so it's going to compare the similarity of all these sequences with our PDB structure which is I think it's a GPR 120 GPCR, and so we could use that as our reference one. So you click on that, and we want information about the similarity in each position of this TM region. So we selected the TM region, and we check this box, and we go OK. This builds a nice table, and so we can see, first of all, it's showing us that obviously 100% similarity. This one has 18% similarity with, so PAR4 has 18% similarity with our template, and adrenergic B GPCR has 80% identity. And so we could also see at position, these are the positions in the alignment that we selected, and it nicely colors the, so you have a table of exactly each residue at each position and how well conserved they are. You can export this table to Excel if you wanted to, and other things. And just wanted to show you the last thing is that you can extract alignments. So if you wanted to extract this TM1, you right click and choose extract selected alignment block here. And so we now have two alignments. We can rename this one to TM1, for example. You can also make a residue number table, which has the residue numbers for your selection. You can also with the labels as well. So you've got the particular, so similar to the similarity thing, you have the labeling at each position. And you can copy the alignment and maybe hide. You can hide that region if you're not interested in seeing it, and change that. Okay. So that's an overview of the alignment features in ICM and sequence features. You can also do a blast search, which we'll be using in 2 weeks' time when we start doing modeling. So to do a blast search, you go to the search tab, choose NCBI blast, enter, first you need to load the sequence. So we go to Uniprot, and the sequence we're interested in is FFAR4_human, which is the model we'll be making next time. You see it's loaded here, and then we can go to blast, NCBI blast, and choose that sequence we just loaded in. And we can choose to do a blast search against all the sequences in the PDB, and maximum hit 100, and then go search. So this does the blast search. This is a good way of finding a template for our GPCR, which we're going to model next time. So it's a good idea to So you can see that we have a hit here. The first hit is has PDB code 4S0V. So the similarity is not great because we know it's a GPCR. So we would need to, you can see that you click here, you can select, you can see how well the coverage of the alignment is to the template as well. So you need to maybe view a few of these to check which one would be the best for the model. And I think if you double click here, that loads the structure. Okay, no it doesn't. It loads the sequence, I think, for that. Yeah, just loads the sequence of this protein. Okay. So I'd like to thank you very much for your attention. If you have any questions after the workshop, after the webinar, please feel free to email. The next one will be in 2 weeks' time on protein structure modeling and structure analysis.